Telomerase activation therapies represent one of the most exciting and potentially transformative frontiers in the field of anti-aging and regenerative medicine. At the heart of this approach lies telomerase, a ribonucleoprotein enzyme complex responsible for maintaining the length of telomeres, the protective end caps of chromosomes that shorten progressively with each cell division. In most somatic, non-reproductive cells, telomerase is largely inactive after early development, leading to gradual telomere attrition that eventually limits cellular replication, a process known as the Hayflick limit. Telomerase activation aims to counteract this attrition, essentially reawakening a dormant mechanism that nature designed only for our earliest stages of growth and for specific cell types such as germ cells and stem cells. The idea of reactivating telomerase in adult somatic cells has profound implications. By restoring the ability of cells to maintain telomere length, scientists envision a therapy that not only slows aging at the cellular level, but could potentially reverse it. Experimental methods for achieving this include gene editing techniques, particularly those targeting the HTRT gene, which encodes the catalytic subunit of telomerase. Upregulating this gene has been shown in preclinical models to extend telomeres, restore cellular vitality, and enhance the function of tissues compromised by age-related degeneration. What makes this area of research particularly promising is the evidence from animal studies. In several experiments involving mice, telomerase reactivation led to rejuvenation of tissues, improved organ function, and extended lifespan, all without a significant increase in cancer incidence under tightly controlled conditions. These findings suggest that under the right parameters, telomerase activation could become a powerful tool to combat diseases of aging such as cardiovascular disease, neurodegeneration, and immune decline, all of which are influenced by telomere dynamics. The use of gene editing technologies such as CRISPR/Cas9 and viral vectors to deliver telomerase activating genes has opened the door to highly targeted, tissue-specific approaches. For instance, therapies could one day be designed to selectively reactivate telomerase in tissues that are most vulnerable to age-related deterioration, like the brain, heart, or bone marrow, while avoiding activation in tissues where uncontrolled cell proliferation would be more risky. This level of precision, while still largely theoretical, is becoming more feasible as our understanding of genomic regulation deepens and delivery systems become more refined. There is also a growing body of interest in small molecule compounds that may indirectly stimulate telomerase activity or enhance its stability. These compounds, often derived from natural products or engineered synthetics, represent a non-invasive, more scalable alternative to gene therapy. Some early candidates have already shown promise in boosting telomerase expression in cell cultures and animal models, offering a potential bridge between highly experimental gene editing approaches and more accessible clinical applications. It is true that one of the main concerns surrounding telomerase activation has been the potential for oncogenesis, since many cancers exploit telomerase to achieve unchecked growth. However, it's important to note that cancer is not caused by telomerase activity alone, but rather by a combination of genetic mutations, disrupted cell cycle control, and impaired immune surveillance. With careful screening, appropriate patient selection, and localized or time-limited therapeutic delivery, the risks of telomerase activation can be minimized, especially when the therapy is aimed at regeneration rather than indefinite proliferation. In fact, some researchers argue that the aging process itself may elevate cancer risk due to genomic instability from critically short telomeres. In this context, restoring telomere length through telomerase activation could actually enhance genomic stability and lower the incidence of age-related cancers by preventing chromosomal degradation and dysfunction. This hypothesis is still being explored but offers a counterpoint to the prevailing caution, suggesting that telomerase may be more of a guardian of cellular integrity than a threat, if used judiciously. Ultimately, telomerase activation therapies hold remarkable promise as a pillar of future regenerative medicine. They represent not merely a cosmetic effort to slow the appearance of aging, but a biological strategy aimed at addressing the root mechanisms of decline and degeneration. While still in the experimental phase, the convergence of gene therapy, molecular biology, and precision medicine is accelerating progress toward making telomerase activation a viable and safe therapeutic option. If successful, such therapies could redefine how we approach aging, enabling people to maintain not just longer lives, but healthier, more productive ones well into old age.